Mikhail, I think we're going to start. Is it on? Okay, you can hear me in there? Okay, good. Oh, but I can't. I got to move this so I can advance my slides. No, I don't. Okay, welcome everybody. Thanks for having so many people here to support Randi. I probably have too many slides to introduce Randi, so if you feel like it's going too long, you just tell me to speed it up. But, one of the things that I love about getting ready to introduce students for their defense is you get to kind of do a deep dive into pictures of them that you've never seen before. And so, I've learned some things about Randi in this process, and I've gotten to know Randi very, very well as a student. But what I know is that Randi started life with very little hair. And Randi now has very little hair. But what I learned is that there was this time in the middle that Randi had hair. A lot of hair. And that was a shocker to me. I've never seen that before. And though short hair or long hair, Randi has always enjoyed being outdoors, being at the beach, exploring things. Short hair Randi, long hair Randi, going on camping trips, hiking trips with people. And also another thing that I already knew about Randi, but I got to see more of, as this picture starts to allude to, is Randi is very metal. In case you didn't know that about her, that is a big part of her life. So let that be a lesson to you guys. If your Facebook pages are open to everybody, your advisors will find the pictures. Randi started as a wee young one with hair at CSUMB. This is a picture of Randi from her very first poster ever. You can see her hair got really short in preparation for becoming a scientist. And, as a student in my lab, I met Randi as a student in my conservation genetics class in spring 2019. She was a student who always came to class, sat in the very front, super excited, super interested, and, really kind of then approached me about getting to do some work in my lab with population genetics. She started, the summer that she started working in my lab, we started this collaboration with Chelsea Wood up at University of Washington. And the idea was that she was going to optimize potential, protocols for working with parasites. Her summer internship started, in June as expected. The parasite samples arrived in September, so you can imagine she didn't really get to do that. Instead, she got to extract some DNA from other things and learn some lab techniques. One of the things, but then Randi graduated in spring of 2020, which meant that she graduated with no graduation ceremony, but having no graduation ceremony means that you can have an amazing photo shoot with microscopes. And then Randi continued on in her studies at Moss Landing Marine Lab, where she stayed working with me, which was such a treasure for me to get to keep Randi as a student. And as a graduate student, she really did a lot of things. She had her own research, she mentored many students, she taught classes, she secured a lot of grants, and she also worked really, really hard outside of Moss Landing. So just as a quick overview, her research focus, she's going to tell you all about her research, so I'm not really going to focus on that, but she worked on parasites. When Randi, in the spring semester of her last year, she was supposed to go on this UROC-funded trip up to University of Washington where she was going to learn to identify parasites. Then, of course, COVID happened. She didn't get to go, but somehow we found the money for her to go her first year of grad school. To do that, she had to take a lot of pictures of parasites, and I gave her my laptop, my iPad, my school lab iPad to take with her so she could take really good pictures, which meant that all of those pictures showed up in my photo stream. And there's a mixture in my photo stream of children and parasites, and I feel like nothing is better than that. So Randi spent a lot of time in the lab, collecting data, extracting DNA from teeny, 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 tiny parasites. And for those of you who have done any DNA extractions, one of the things I did genetics for my thesis, and I had pieces of tissue that were really small, but it meant that every time I did a DNA extraction, I could have done 100 of the same tissue sample, whereas for Randi's thesis, it's a one and done. You do one extraction, you use the whole organism, and there's no redo after that. In addition to the work that Randi did as part of her thesis, she was also involved with some larger projects with Dr. Eric Crandall, who died recently. And she was part of 2 publications as part of that. including one in PNAS. So like the student has way surpassed the master. I will never publish in PNAS, and that is totally fine with me. And then as a teacher at CSUMB, she taught intro bio labs, intro bio discussions, and she also helped me teach a spring break crash course to give students who lost the ability to get to do hands-on molecular tools during COVID. We taught a crash course over spring break to give students more hands-on molecular tools. And she also was nominated for the CSU Biotechnology Student Teaching Award. I think they made a mistake when they didn't give it to her, but she was nominated. She also mentored many, many students as part of this program. So Randi was part of the research internship and molecular ecology program, where we had 5 CSUMB students and one REU student from the University of Puerto Rico. And so we had an amazing summer where Randi helped mentor all of these students to learn to do molecular tools and do, look at sequences and do an analysis of sequences. And here's some of our students presenting their posters. Randi also was supported in some ways by the NSF grant that Chelsea Wood at UW and Stuart Sandin at Scripps are part of, but really that only covered her research costs. It didn't fund her as a graduate student. So she collected many disparate grants along the way. She got CSUMB TA-ships, where that included tuition waivers, the Myers Trust Award, the Moss Landing Student Body Wave Award, the CSUMB Research and Scholarship and Creative Competition Grant, the Moss Landing Scholar Award, the Coast Student Travel Award, the Moss Landing First Generation Scholarship, and the CSUMB Biotechnology Faculty Graduate Student Research Award. So she had many awards along the way that helped fund her as a graduate student. And I think a lot of graduate students don't wind up having this level of success and experience writing grants as their graduate student. Outside of Moss Landing, like many Moss Landing students, Randi worked a lot outside of her thesis, and it's amazing that she has finished in this time given how much she was working outside of Moss Landing. She worked at Granite Canyon Marine Biological Lab, where she had a lot of great support from her community there, most of whom are here. I like to take credit for that she continued to do field work and love of algae. And so while she was there, she did lots of environmental consulting work, getting very dirty, doing lab work, and having a lot of fun with her peers and her colleagues there. And I think that's one of the things that I've seen about Randi is that she's built such great community among the undergraduates, other graduate students, her colleagues, everybody that she works with, and I think that allows her to have such great support, both within academics and outside academics, from CATS, from family. And to me, it's no surprise that, you'll see in a second the amazing outfit that Randi is wearing, but it's no surprise to me how goofy she is given, what a goofy level of support she has from her family. So with that, I'd like to welcome Randi up to talk to you about her research.
Oh my gosh. I did not expect a lot of those pictures to see the light of day. Hold on, I can't get my mouse. All right. Okay. Thank you guys so much for coming today. It really means a lot to me to see so many of you out in the audience. Like Allison said, I've worked really hard for the past 4 years to get through this degree, so thank you so much. I'm so excited to talk about what I've been doing. I've been using population genetic analysis to better understand how dispersal capacity works in trematodes, which happen to be 2 different families, which I'll get into. Okay, so in marine and terrestrial systems, life history drives the distribution of organisms and it informs population connectivity. So most marine fishes and invertebrates have a bipartite life cycle. So that means they begin with a larval phase that is their main dispersive phase, and then they end in an adult phase that is partially or completely sedentary. And this dispersive larval phase is really key in driving gene flow, and it determines subsequent population and community dynamics. How organisms disperse is also a crucial aspect in how the species will evolve and how they may persist when faced with environmental change and other human impacts. Now, there are many studies that focus on the dispersal of marine fishes and other invertebrates, and often you can find multiple studies covering the same species of fish or invertebrate, but that same breadth and depth of research just does not exist for the parasite taxa. Which is really interesting because parasites have even more complicated life histories than a lot of marine fishes and invertebrates. They have dispersive larval phases, just like the others, but they also utilize other hosts that could potentially increase their dispersal for them. And the complexity of their life cycles varies widely across the parasite taxa. And because there's such an array of life histories amongst the parasite taxa, we can group them into two major categories. So there are the parasites that are direct transmitters. This means that they only use one host species to complete their life cycle and can only move between conspecifics. And then there are those that have complex life cycle transmission. So this is the obligate use of multiple host species. And it's generally thought that parasite dispersal and therefore population genetic structure is driven by their hosts, and that differences in life cycle complexity can lead to differences in parasite dispersal capacity. So a parasite's dispersal capacity will probably range from low to high dispersal based on certain life cycle characteristics. The first major one is life cycle complexity. That's the minimum number of host species that is required for the parasite to complete its life cycle. And then there's host specificity. That's the number of host species that can be used by a parasite at a given stage of their life cycle. And then, of course, it matters what types of hosts they're infecting. Are they infecting more sedentary hosts? Are they infecting hosts that can swim a lot further? So parasites that utilize direct transmission or have narrow host specificity or who utilize more sedentary hosts will likely have a lower dispersal capacity than those parasites that have complex life cycle transmission who utilize broader host specificity or more agile hosts or hosts that can swim a lot further. Okay, I cannot give this talk without presenting a gross parasite slide, so I'm sorry. But some of you might be thinking, why should I care about how parasites are moving? They're disgusting, get them away from me. And which is fair, because we often associate them with disease, infection, and as a general pest to human existence. But what can I say? Nature is indeed metal, and you know how I love metal. Like it or not, parasites are just a part of the ecosystem as any other organism. They are part of that overall balance. Unfortunately, parasites have often historically been overlooked in terms of their contributions to community dynamics. And so there are a ton of knowledge gaps that have persisted throughout parasite ecology. But parasites' presence and interactions within all systems just cannot be understated. For example, Morton et al. In 2021 did a food web study in the Channel Islands and built one food web with no parasites. So this is just predator and prey interactions in the blue. Then they made a food web adding parasites in the red. That nearly doubles the total number of species interactions. And as more of these studies come out, we're starting to see that parasitism is often more abundant than even predator and prey interactions. And so parasites have a very close relationship with their hosts. I mean, parasitism is a type of symbiosis, and therefore it can affect multiple things throughout the environment. Parasites can alter host behavior. They can change their phenotypes. They can limit their growth. They can change their reproductive timing. And parasites are also a significant driver of disease, hence that last slide. But it's actually a form of host population regulation. Kind of like how predators help mediate the abundance of prey animals, parasites do the same thing for their hosts. And so therefore, parasites also impact the nutrient cycling. They're part of that overall system. And so the removal of parasites, or even the addition of parasites to a system, can have cascading trophic effects. You can imagine if you just removed all the parasites, that's going to affect a lot of other interactions happening in the web. And so this undeniable connection and effect that parasites have with their hosts and therefore their environment indicate that understanding how parasites move is essential to our understanding of population dynamics, population and community dynamics, especially in the face of climate change and other human impacts. So as I said earlier, parasite population genetic structure is generally assumed to be driven by that of their hosts, but we need more empirical studies on wild populations to better understand parasite dispersal and also to better understand parasite responses to environmental change. So islands are a really great way to study parasite dispersal. For example, if you're a parasite that lives in a reef fish, that reef fish is not going to be able to swim the hundreds of kilometers across an island system. It's probably going to stick to its home island. So you as a parasite will just get to disperse your eggs around that home island. But if you're a parasite on a shark, that shark has the capacity to swim circles around the island system, and so you get to spread your eggs a lot further, which increases your dispersal capacity. So island systems provide a really great way for us to analyze parasite dispersal, because if populations of parasites across the islands have similar genetic structure, that means they're somehow traveling that distance. So this leads me to my research questions. First, I want to look at what is the population structure of adult and larval trematodes sampled from islands in the Northern Line Islands and French Polynesia. And then I want to estimate the dispersal distance of the trematodes in these systems. So my study is focused on 2 island systems within the Central Pacific. The first is the Northern Line Islands. That is located about 1,600 kilometers south of Hawaii, just above the equator. And then French Polynesia is located about another 2,000 kilometers south of the Northern Line Islands. And so there were multiple expeditions to these islands that I was not a part of, unfortunately. Someone wanted to take me to Tahiti. And the reef fish were caught at the forereef of each island and brought back to the lab for analysis. So those fish were brought back to the wood lab at the University of Washington, and they were dissected layer by layer, especially the intestinal and muscular walls, looking for parasites using microscopy. The main host that we have is the Acanthurus nigricans. That's just a type of reef fish. And then, all the adult trematodes were found from Acanthurus nigricans, and the larval samples were found from the Stochastis species. So those parasites, once they were found, they were put into vouchers and sent to CSUMB in 2022, where I had control of them. And due to the lack of reference literature surrounding parasites, I spent a lot of time optimizing the laboratory protocol to even get DNA out of them. But once I got our extraction protocol optimized, we extracted DNA from the parasites and targeted the mitochondrial CO1 locus. The CO1 gene is commonly used in phylogeographic and population genetic studies. It accumulates mutations fast enough for us to identify between closely related species, but it's also a conserved gene, so it's a conserved gene between conspecifics, so we're able to utilize that for population genetic analysis. So once we got the DNA samples, we sent them off for Sanger sequencing. And I do want to highlight, Allison talked about this a little bit, but I wanted to highlight just how much help I had during this process and how difficult it was. So part of this work, I got to mentor 6 undergrads during the RIME program. And these undergrads went through hundreds of parasite extractions and a countless number of PCR. And they were able to use the data for their own independent research projects, which is really cool. And for a lot of these students, it was their first research experience ever, let alone molecular experience. And when I say that the protocols that I brought to them were not simple by any means, I really mean it. Firstly, these trematodes were very small. I don't even know if you can see them up there. But that adds a layer of difficulty because when you start the process, you have to start by keeping track of things you can barely see. And additionally, their small size means you have to use a whole organism for DNA extraction, as Allison mentioned. And in a lot of other genetic research, you can just take a tissue subsample, but not for us. So if the extraction or PCR did not work, you often lost that sample. And if you didn't lose the sample entirely, a lot of times that low tissue size just led to bad quality extractions, which led to very messy and unusable sequences. So I stared at a lot of squiggles during this process. Makes your eyes go crazy, but this happened a lot. And so we couldn't rerun a lot of extractions, but often we could go back and try PCR again if the sequences were bad and try again. But all of this process was extremely time-consuming, tedious, and frustrating. And I feel like these 2 pictures really sum up the emotional experience of that work. So after a lot of mental pain, I was finally able to accumulate enough good quality sequences to use for my analysis. So I uploaded the raw sequence data into Genius, which is a bioinformatics software, and I cleaned all the sequences, and then all of my further analyses were based on multiple sequence alignments that I ran in the software. So going back to my research questions, remember how I said, oh, I'm gonna look at the population structure of trematodes. And I thought it was gonna be 1 trematode species across all the islands. Turns out there was 3, which means I actually had 3 questions for my first question. Things got kind of complicated. Firstly, there were larval samples collected from each region, but they lack that morphological characteristics needed for species identification. So we needed to first figure out who they were to be able to do population genetic structure on them and if they could be included with the adults or not. And then there was 1 species of adult trematode in the Northern Line Islands and 1 that was different in the French Polynesian Islands. So in the Northern Line Islands, we had family Microscaphididae. Throughout this talk, I'll refer to them as the microscaphidids or the Northern Line Island adults. And then in French Polynesia, we had family Paramphistomatidae, or the Paramphistomes, and I will refer to those as the French Polynesian adults as well. So these were the larval samples, and as you can see, they just don't have a lot of characteristics to identify them by, so that's why we first needed to figure out who they were. So first I asked, are the larval samples related to the microscaphidids? This is water, I promise. So first, a neighbor joining consensus tree showed that all of the larval samples formed in different clades from the Northern Island adults. The Northern Island adults are in the black, the larval samples are in the red and blue, so already you can see a little bit of separation between the two. To further visualize this relationship between sequences, I made a haplotype network. And so you can kind of think as haplotypes as sequence variants, or how similar are these sequences to each other. So the more samples that share the same haplotype, the bigger the circle. And the colorless circles in between the colored circles are the number of mutational steps it takes to get to the next haplotype. So here you can see that all the larval samples, again in the red and blue, are not connected to the Northern Island adults in the black, which suggests that the sequences are probably different from each other. So then I did phi statistics, and phi statistics is a method of measuring the amount of variance among the samples that can be attributed to any differences among the populations. So a phi CT of 0 means that the populations are completely mixed. A phi CT of 1 means that they are completely separate. And researchers often use phi statistics as kind of like a proxy for speciation. So a phi CT of 1 suggests different species. So in this case, no matter how the larval samples were grouped by region and compared to the Northern Island adults, there was always high significant genetic structure. So this further suggested that they are different from each other. And the next analysis I'll be showing you is barcoding gap analysis. This analysis is used in every question from here on out, so I just want to orient you to the figures you'll be seeing. Barcoding gap analysis is a method for delimiting species, that's what I was trying to say. Basically, if you have two groups of individuals, you can compare the interspecific variation of the group to the interspecific variation between groups. And for a group of sequences that come from the same species, you would expect interspecific variation, or the genetic distance between sequences, to be very small. And if they're different species, the intraspecific genetic distances are then expected to be smaller by at least an order of magnitude than the interspecific variation. And this leads to what's known as a barcoding gap. And the size of the barcoding gap can give us clues as to how closely those two groups may be related. And if they're the same species, the intraspecific and interspecific variation will overlap. So that blue and yellow will overlap on each other in the figures you'll see. So first, I compared the interspecific variation within the Northern Island adults to the interspecific variation between the Northern Island adults and all larval samples. And what I saw was that the interspecific variation was much greater than the intraspecific variation. And it led to a barcoding gap of about 30%. And this indicates that the larval samples are different from the Northern Island adults. So with all that evidence put together, are the larval samples related to the microscaphidids? No, they're definitely not. But then I had to ask the same question for the paramphistomes. So immediately I saw kind of the opposite. The larval samples in the red and blue were starting to group within the clade of the French Polynesian adults. Except there were 5 unidentified Northern Island larval samples that kind of hung out on their own separate clade. And you can see that in the haplotype network as well. So the French Polynesian adults are in the gray, the red and blue is the larval samples, and you can see that the larval samples all group with the French Polynesian adults, except for, again, for those 5 unidentified random little red dots hanging out. And you might notice that there's a big separation between the two clades, or between the French Polynesian adults here, and I'll get into that later, but for now, what's important is that the larval samples are grouping with the French Polynesian adults. So when I ran phi statistics, I saw low significant genetic structure between larval samples and the French Polynesian adults, and I saw no genetic structure between the French Polynesian adults and larval samples. I did, when I ran just the Northern Island larval samples with the French Polynesian adults, I did have quite high genetic structure, but those, again, it's attributed to those 5 just unidentified larval samples. So this suggests that the larval samples are similar to the French Polynesian adults. So then when I did barcoding gap analysis, you can see that the interspecific variation within the French Polynesian adults is nearly identical to the interspecific variation between adult and larval samples. Again, there's that little outlier. Those are those 5 weird samples from the larval side. So all of this together, are the larval samples related to the paramphistomes? Yes, they are, except for those 5 weird ones. So I figured out who the larval samples were, and I removed all of the larval samples that were associated with the Northern Islands because I wasn't going to use them, and I was able to group the adult and larval together for the paramphistomes. So now that I knew who they were, I could move on to the next set of questions. Microscaphidids and paramphistomes both have are trematodes that both have complex life cycles. So that means there's more opportunities for dispersal. However, all these hosts are sedentary hosts, so this limits the potential dispersal distance because they can't go that far. So if we go back to our dispersal capacity scale, due to these life cycle characteristics, I expect these trematodes to both fall somewhere between low to medium dispersal capacity. So let's start with the analysis for the Northern Island adults. So after removing the larval samples, I had 83 microscaphidids across 3 islands, Palmyra, Turena, and Christmas Island. This is another neighbor joining consensus tree. Here the nodes are labeled with the number of nucleotide substitutions per site. And so what happened in this tree is that samples from each island tended to group with samples from that same island, except samples from Turena tended to group in a separate clade from the rest of the samples. However, that clade difference is less than 1 nucleotide substitutions per site. So then I ran phi statistics, and what I did was I first ran all sequences together grouped into one population, and when all the sequences are considered one population, it showed strong differentiation within the population. And so this means it's not a completely mixed population, but it's also not completely separate. So then I calculated population pairwise phi-ST comparisons. And here again, I saw a high genetic structure between all the populations, which indicated low gene flow across the islands. Christmas Island and Palmyra were the most closely related, but not by much, which is interesting considering they're the furthest apart from each other. And because there was such strong genetic variation in the Northern Islands, I wanted to visualize population connectivity, so I made another haplotype network. What we saw was that this haplotype network suggests that the populations are still genetically connected even though there's such strong differentiation. And the absence of reticulation between nodes indicates that there's low gene flow. So what that means is node 3, you can see all those haplotypes expanding off that central node. If those haplotypes were interfacing with the same thing from another major node, that would indicate high gene flow, but that's not happening here. And so then I wanted to look at the distribution of haplotypes across each population in the Northern Islands. Here, the white slices are unique haplotypes to that specific island. The red is the shared haplotype across the island. So there was only one shared haplotype, but that means that there is still some level of gene flow happening between the islands. It either happened just at some point in time, or it's just very little, like enough to keep them from speciating. So for the adult microscaphidids across the Northern Islands, there was very strong genetic structure, which means minimum gene flow and limited dispersal. And that limited dispersal is likely due to their hosts. However, there's still that level of connection we still saw. So what are other mechanisms for that limited gene flow? And we can look at surface currents in that region. This is a screenshot of the surface currents around the Northern Islands. The red arrow is the location of Christmas Island. The black arrow is the direction of currents that are in the equatorial current. And the Northern Islands are located like right above the equator, so that's where a lot more stronger and concentrated currents are flowing. And so this could increase the number of eddies and fronts that potentially bring parasite larvae to the next island. And other studies have modeled larval connectivity in this region. Mary et al. In 2017 simulated the passive movement of fish larvae across the Northern Islands, and what they found was that Palmyra and other Northern Islands received about 3 to 11% of its total particles from more southern islands. So this shows that larvae could potentially move across this region. Microscaphidids, though, produce a lot of short-lived larvae. So surviving that long trek across the islands is probably difficult. However, these microscaphidids are hermaphrodites. So that means that only one of them needs to get to the island to effectively colonize that island. Whoops. Sorry. So it's possible that surface currents can transport larvae between islands, allowing for some level of gene flow. And it keeps the populations from speciating, but a high mortality rate reduces the number of individuals that make it to adulthood, thus limiting gene flow. And this can help explain why we're still seeing a little bit of genetic or population connectivity between the islands, despite the microscaphidids having such sedentary hosts. Okay, so coming back to kind of our roadmap of questions, we have identified high population structure in the microscaphidids across the Northern Islands, and we can move on to the paramphistums. Except, turns out there was actually cryptic species present. So what turned into one question kind of got a little changed just because there were cryptic species. It was just another plot twist in this long saga, so yay me. So for the paramphistums, with the adult and larval samples, I had 132 trematodes across 5 islands. And so this phylogenetic tree was like the first indication that something was weird, because there was these 2 distinct clades starting to form, between like a clear split between all samples. And so if you remember this figure from earlier, where I was comparing the genetic variation between the French Polynesian adults and the larval samples, you can see this clear gap between the intraspecific variation within the French Polynesian adults, and high intraspecific variation can indicate the presence of cryptic species. So also that was kind of my first indication that something weird was going on. So when I put it into a haplotype network, that high intraspecific variation was further emphasized in the network because you can see 2 distinct clades forming. Each clade had samples from every island except for Raiatea. Raiatea was reserved to just clade A. So then what I did was I grouped the sequences by which clade they fell into in the haplotype network, and I calculated phi statistics. So when comparing clade A to clade B, the phi CT was very high, 0.85. That indicates that they were definitely different from each other. And when they were considered their own populations, clade A and clade B had pretty significant high genetic structure as well. So other studies suggest that interspecific variation greater than 5% between individuals of the same species suggests cryptic species. And when I did a barcoding gap analysis of clade A versus clade B, what I saw was that the mean interspecific variation was about 8.85%. And this kind of falls into what other studies are seeing for other Degenian trematodes when they identify cryptic species. They also get a range of interspecific variation from about 5 to 10%. So then I just wanted to compare, or put this in the context of some other data that I had. I wanted to compare the genetic variation between 2 groups that I knew for sure were different families, which was the paramphistums and the microscaphidids. And so when I did a barcoding gap analysis, I saw a mean interspecific variation between the 2 adult families of 32%. And 32% is much higher than that 8% we're seeing in clade A and clade B. So what that tells me is that clade A and clade B are likely probably the same family. They're probably both still paramphistums, and they're probably closely related, but they just didn't have any identifying characteristics to see them just based on morphology. So even though we found cryptic species in the paramphistums for French Polynesia, we still found evidence for high genetic structure, low gene flow in the French Polynesian islands as well. So now I can actually move on to my second question, my real second question, which is what is the estimated dispersal distance of the trematodes in these systems? And so now that we had population genetic structure information for both the microscaphidids and the paramphistums, we could try to estimate the dispersal distance by using isolation by distance plots. So these plots compare geographic distance to genetic distance. And if there's a significant relationship between the two, we can use the slope of the line to calculate a dispersal width. So I basically made isolation by distance plots for every parasite group across every region. I even decided to group the cryptic species together just to see what would happen. And so the isolation by distance plot for the microscaphidids in the Northern Line Islands showed no significant relationship between genetic and geographic distance, which means no dispersal width could be calculated for this system. Perhaps if we had more islands in this data set, more samples from more islands in this data set, we may have seen a relationship, but we don't have that, so we don't know. So no dispersal width could be estimated. Out of all the paramphistome isolation by distance plots, only clade A had a significant relationship between genetic and geographic distance. And so that means as geographic distance increases, the genetic distance also increases. And so I can use this to calculate dispersal distance. And so Roussey's equation is how you do that. You basically take the slope of the line and an effective density, put it into the equation, and you get dispersal distance. Easy. No, there's no census data for trematodes. Definitely not effective density data out there for these paramphistums specifically. So I had to do a little workaround. I had to combine 3 different sources of data to get an effective density for paramphistums. The first thing I did was I looked at the host density data for Acanthurus nigricans. I was able to find a paper on the density in the Northern Line Islands. So this is where I make my first assumption. I assume that the Cantharus nigricans density is probably similar enough between the Northern Islands and the French Polynesian Islands because they are such similar systems that I can just use that. But that is my first assumption. And then I calculated host infection rate. So basically, I went through our data set of a Cantharus nigricans fish and looked at who of those fish had at least one paramphystome present. And so then I calculated the host infection rate that way. And then the other thing I had to contend with is that the number of trematodes per fish can vary super widely. One fish can have anywhere from like 1 to 500 parasites. So what I decided as to kind of estimate this was I took the mean and median number of trematodes per fish in the data set that we had to create like a range of effective densities based on what we had found. And so the host density data was about 0.1 fish individual per meter squared. My host infection rate was 34%. And it turns out the median and mean were pretty close to each other, 3 and 3.72. So it didn't give us that big of a range of effective density, but still, use it anyway. So when you plug those numbers into our equation, we get a dispersal distance of about 24 to 27 meters. And because of the many assumptions I had to take to calculate effective density, I decided to see how much the dispersal distance would change if I increased and decreased the effective density by like an order of magnitude in both directions. And when I did that, I got a dispersal distance of about 8 to 85. So it didn't actually change like the overall impact of the dispersal distance, it just broadened it a little bit. But if we stick to the dispersal distance estimate of about 8 to 85 meters, that is a small distance range. It kind of barely covers the distance between the French Polynesian Islands, which actually makes sense for what we're seeing in the paramphystome genetic structure, because if they do rely on their host's range for a majority of their dispersal, we remember those hosts are really sedentary, so they can't traverse that island or that distance between the islands very easily. And so the trematode eggs probably just drift within the local environment, very slowly dispersing throughout the whole system, which is low gene flow. So that's exactly what we saw in our genetic structure for clade A, which was a phi-ST of about almost 0.5. So we expected the dispersal capacity of microscaphidins and paraphystomes to be somewhere from low to medium dispersal. What I actually found was much lower dispersal capacity than I had expected. So even though there were cryptic species present within the paramphystomes, I was still able to see evidence of low dispersal, and it also reflected in that small dispersal distance that I calculated. So something else that this data helps support is some other hypotheses that are present in parasite ecology. The trematodes used in this study are autogenic parasites. So this means that all of their hosts live in one environmental medium, so they're all marine hosts. There are also allogenic parasites. So these are parasites that have hosts from different environmental mediums. So you can imagine if you're an allogenic parasite, you should have higher dispersal capacity than an autogenic parasite, because you could have a bird take you a lot further than maybe other fish could. So my study provides support for the idea that autogenic parasites in general possess low gene flow and high population subdivision. And so obviously this wasn't a comparative study, but it does support the idea of low dispersal in autogenic species. And this data is coming from a natural system in the Central Pacific in marine trematodes. And so other studies have looked at this in other systems, but to my knowledge, none of the data has been from my exact system, which is pretty cool. So environmental change and human impacts such as fishing continue to drive biodiversity loss, which is a direct impact of parasite populations. They rely on those biodiversity to survive. And so knowing more about how parasites disperse allow us to make inferences about how their populations will be impacted by losses. So in general, it's theorized that having low dispersal results in a higher susceptibility to fishing pressure, because you don't have as many options to persist or to continue your life cycle. But trematodes have a complex life cycle, and so they may be able to persist through the fishing pressure by utilizing their first intermediate hosts, which are gastropods. They can kind of utilize them potentially as a sanctuary, where they can kind of wait out the fluctuations in host abundance and make it through fishing pressure. And in fact, Wood et al. In 2014 found that microscaphidid abundance was actually higher on fished islands than unfished islands. So even though I found low dispersal in microscaphidids, their specific life history allows them to potentially persist through fishing pressure. And so this highlights that collecting more data on natural populations of parasites allow us to make more nuanced inferences about parasite persistence as environmental change and human impacts continue. So there are complications of the study extend well throughout parasitology. There are multiple times when ensuring efficient sampling size became an issue. In general, it's just difficult to plan out how a parasite study because you don't really know how many parasites you're going to find. You can collect as many fish as you think you need, but it still is kind of left up to chance as to who's going to actually be there when you get all the way back to the lab. And for me, the sampling was done prior to my research, so there was really no way for me to go back and collect more samples. And there was also the problem of major gaps in biodiversity knowledge within parasitology. Not all parasites have identifying morphological characteristics, and so when you conduct a population genetic study, all of a sudden you're faced with cryptic species that you didn't know were present, just like what happened to me. So that lack of biodiversity knowledge leads to difficulties establishing laboratory protocols as well, because there are often no reference genomes or lab protocols that other people have already figured out, so you have to go through that optimization period that I mentioned. And to do this, you have to utilize some of those precious samples. So then it takes you back to the sufficient sampling thing. Then you might lose some samples as you're trying to figure out that optimization period. And as I mentioned earlier in the talk, the actual data collection process also leads to a loss of samples due to the small tissue sizes you're working with. So a lot of times, parasite genetic work is just like this big catch-22 that's cycling around. But on the flip side, my research has been able to also provide some answers for those who might be doing other parasite research. For example, I was actually able to piece together a DNA extraction and PCR protocol that did work, so others could use that protocol. And additionally, all the sequences used in this study have been deposited into the NCBI sequence archive. So other researchers can use those sequences for their phylogenies. Maybe it will help resolve parasite phylogenetic trees, or increase our understanding of overall parasite biodiversity. So that's something I'm definitely excited by. So overall, life history and transmission strategy can inform our understanding and estimates of parasite dispersal. However, other factors must be considered, right? There are those geographic barriers that I mentioned, like distance between the islands themselves. There are other anthropogenic factors like fishing that produce pressure outside of their normal host environment. There are physical processes such as surface currents and eddies, and other biological interactions like competition between species, parasite sexual mode. All of these things are part of the balance of understanding parasite dispersal. It's very complicated. So for future research, things I'm really excited by and want to see in the future is I want to be able to compare population structure of parasites with different life histories. So maybe some that have direct life cycles, and it would be cool to do it across these islands as well. I also want to expand the analysis with the addition of genomic data. We're actually starting that collaboration with some researchers at Hasselt University because we're only looking at CO1, right? That's a small region. It can tell us some things, but it can't tell us everything. So if we can add genomic data to that, it would be really cool to either see how it corroborates what I found or maybe changes what we found. I'm very excited by seeing the outcome of that. So I have a lot of people to thank, and I just want to get through that first. First, I need to thank all of my funding for the research and also, my hours. That really helps with this research a lot. I really have to thank my committee. Allison helped, my advisor. Without you, I literally would not be standing here. The amount of times I have just texted you in the middle of the night, freaking out, thinking that my thesis was done for. And we made a plan, we made a meeting, and I always say that I always leave your office feeling a lot better than when I came in. I even came to her office one time and said, I'm going to quit if we don't figure out something. And we figured out something. Allison is amazing, so thank you so much. I also want to thank Amanda Kahn, my co-advisor. Thank you so much for letting me use your lab space, for helping me with my writing, and also for teaching molecular techniques. That really helped me be able to lead an independent research project and help me build my protocol as well. I would like to thank Chelsea Wood. She's a parasitologist at University of Washington. She's on Zoom. Hi, Chelsea. Thank you for trusting me with all your parasite samples and also for just opening the door of parasites to me. I never thought I would be interested in parasites, but they're so weird, and that's what I love about them because I'm weird. They're so cool. So thank you so much, Chelsea, and your help. And anytime I had a question, you were just right there, helping me, get through, just see through the weirdness, really. Sarah, thank you so much. Also, your molecular techniques class, you co-taught that with Amanda. That really helped me figure out my protocol. And thank you for letting me use your lab space and just for the overall support, you guys. I just also want to shout out that I have an all-women committee. That's so cool. It's happening more and more, but it still doesn't happen that often, so that's just really special. I also have to thank the Sandin Lab at Scripps for doing a lot of the fish collections, or all the fish collections. Katie Leslie and the Wood Lab at the University of Washington for all the fish dissections and parasite identification, for teaching me how to look through a microscope and actually be able to see if it was a parasite or if it was a piece of dust. So thank you. You taught me a lot of what I know. Johanna Weston for some lab protocol guidance when I was despairing about the protocols not working. You validated everything, and because you also had the same issues, and that just really put me at ease, so thank you so much. Nicole Mentova at the Hessel University, she also gave me some data analysis guidance. All the barcoding gap analysis, she was the reason that I found out about that. She showed me how to do it. And you also really validated my experience working with parasites and made me know that it wasn't just me. So thank you so much. I have to thank the RHYME program, all you undergrads who put through that torture that I mentioned. Thank you so much. Without you guys, I wouldn't have had the foundation I needed to do this research. So you guys are awesome. Love you guys. I have to thank all my Moss peers. You guys were I started grad school in the pandemic when I had no interaction. It was all online, and I thought I was going crazy. I thought something was wrong with me, and I wanted to quit school. But when we got back in person, and I started having that peer-to-peer interaction and the teaching that happens between peers, that I realized that was what I was missing. You guys are some of the only people that understand the experience that I went through fully, and you got through a lot of complaining probably from my end. So thank you so much for being there with me. I also have to thank my family and my partner. I would not be here without my mom and dad. I'm gonna cry. They sacrifice everything for me to be here, so thank you. And my partner, I cried so much to you about this. And I knew both all my family and my partner, you guys didn't really know what was going on, but you guys were so supportive. So thank you so much for being there with me. Same with my friends. You guys probably heard the most complaining from me. I really appreciate you being there and supporting me. And again, you guys were the people that lifted me up in times where I didn't think I could do this, so thank you so much. I have to thank Granite Canyon. You guys have been so patient while I've been finishing this, and also just you guys all understand the academic trials. So it was really nice to talk to you guys, and I'm very excited for the next steps moving into Granite Canyon as hopefully an academic. I'm a staff research associate right now. I want to move into the academic pathway. We're already talking about papers we want to write, and my bosses have been so nice to wait for me to defend, to put that on me. So they've been very patient. Thank you so much. And of course, my cats. Emotional support, so thank you so much. They really sometimes were the only thing that got me through. And thank you all for listening. I couldn't not show you a really cute parasite before I leave. So any questions? Thank you.
So people on Zoom can also hear questions. If you have questions, I'm going to bring the microphone to you. So questions for Randi.
Great job, Randi.
Thanks, Sarah.
I'm curious what you ended up finding out was the modification that was needed for the DNA extraction that made things work.
Yeah. So I luckily found a paper that had some primers that it was 3 primers. I was doing 2 sets of primers, but it was a set of 3 primers with an internal primer. And I have an email. I asked you guys how to use 3 primers. I had never done that. And so, you guys helped me figure that out. I did that. And it ended up being a 2-step PCR interaction, which was fun for the undergrads to go through. But yeah, it was just finding the right set of primers, which was really difficult, but also I did a hot shot, or not a hot shot, hot start method for the PCR to find the right temperature. And it just ended up working out. I think also we kind of did a rinse step with the parasites as well, because they came from fish and they're so small, they had like fish tissue residue. I don't know how much it helped, but we did like do like an hour rinse step and just autoclaved like DI water before we moved on to the extraction period.
Awesome. Good job figuring it out.
Thank you.
Other questions?
Thank you for a great talk. I'm really curious, do you think that the dispersal distance has anything to do with the health and well-being of the things that are dispersing your parasites?
Yeah, definitely. I think it definitely has to do with the health and well-being of the hosts. Something that I learned throughout this weird world of parasites is host immune systems actually have a lot to do with sometimes even allowing the infection to exist. They can stop infection, so it's not just the parasites taking over, right? The host also has ways to, fight against it. So sometimes they can, stop that dispersal, or the infection taking over. And then also co-infection of parasites. So if there are multiple parasites in a fish, that fish can get too sick, right? Because these parasites are also competing with each other for resources, and they're trying to win over each other, right? So a host could get too sick and die off. But yeah, it's all about that balance. I mean, it all works out in the grand scheme of the ecosystem. So I definitely think that it can affect dispersal distance just depending on what kind of variables are going into it. Yeah.
Thanks, Randy.
Thank
that
you.
was really great.
Thanks, Amanda.
I'm interested, and I know I got to have like a peek a little deeper because I got to read your thesis too, but about the island biogeography theory.
Yeah.
And I can see where there are ways that parasite-host interactions fit into that, but I'm curious about where you might see that it doesn't fit so well. Like, where are the violations to island biogeography theory, and what do you think's going on that's changing things for the parasite-host interaction?
That's a good question. I think so. I think that the host interactions definitely, like you said, move forward that island biogeographic theory, where the hosts, especially with the parasites that are dispersing so low, like the paramphistomes, who had such low dispersal in that environment, that's kind of how they locally disperse. I think what could disrupt that, I think fishing, for sure. Even though that some parasites can persist through fishing, I think the removal of parasites kind of causes potentially more parasites per fish, which could change like what am I trying to say? It could change the way parasites are dispersing across hosts, where it's not as like what I know with like island biogeographic theory, where there's like a stepping stone model, right? They get here, they get to here, they get to here. It might be even more complicated than that, because if there's more parasites spread out amongst less fish, it could speed up that process. I guess that's what I'm trying to say. Yeah.
Thank you.
Yeah. That was a hard one. I haven't thought about that.
Hi, that was awesome. So I'm really curious about the life history of these guys, and you mentioned that the larval stages can like stay in the snails. Are they actually able to like pause on moving forward in their life history, or is it just like, could you elaborate on that?
Yeah, parasites are really weird. And I just want to throw this out there. This acanthocephalin, they actually are able to, so if they get eaten by the wrong thing, they can actually know that, and they're able to halt their continuation into the next life stage. We don't really know how or why they're able to do this, but they are able to kind of just like hunker down and wait, and hopefully the host that they are in will actually get eaten by the right thing. And sometimes when it does, it actually triggers it to move to the next life stage. Yeah, a lot of these like little interactions between, or these cues, these cues between how parasites know to move to the next life stage is still like a huge area of research. There's like chemical cues, hormonal cues, temperature of the host. So if they are able to wait, I don't know, it might just be that they're not making the choice to wait in the gastropod. It's just that they, because there's less fish, they kind of more of them are just sticking around in the snails, but they're surviving. So then when they do get into the fish host, that's how they are able to persist, Yeah. Yeah.
Any more questions?
Super fun presentation, Randy. Thanks. I'm trying to wrap my head around the life history of these trematodes, right? Because the adults were in the surgeonfish. Larvae, at least of that one, seems to be the same thing, right? Was in the damselfish, and they're both herbivores.
Yep.
So I'm trying to figure out, like, how the heck are they moving from one to the other, and is there a missing link somewhere in there, or something crazy going on?
Yeah, something crazy going on, that's the answer. So you actually brought up a part that I didn't talk about in this talk. There was this whole fiasco about the larval samples, even why were they even present in the reef fish? Because, yeah, like you said, if there are larval trematodes present in the reef fish, that means that they're not at their final definitive host. So for a while, we actually thought the larval samples should be a different species, because if they're in the same host, that means that must be a different species. They can go on to potentially infect more fragile host species. So when we started this project, we were thinking they were different, so that's what we went into. When they weren't, when they were the same, I kind of looked into figure out how they even get there. And when I looked at the raw data set, all of those larval samples on Stegastus were not inside, they were not internal. They were like on the eyes, the gills, the fins. And then I looked into Paramphistom life history, and I didn't talk about this here because it just complicated things, but when they get into their metasecarial phase, so that's where they kind of hunker down and go through a lot of life stages, and that's what those larval stages are. That usually happens within a host, but Paramphistomes, they actually can insist anywhere. So they can just insist on debris, they can insist on algae, and so it's possible. What's probably happening is those larval samples just got there by accident, and that's how we found them. Yeah. So normally it would get on like algae. Those fish would eat the algae, and they would get infected that way. Yeah.
Also, great talk. I was also curious about the life history of these parasites.
Yeah.
And you mentioned like you've seen up to 500 on a single fish. Like, is there a sort of limit on how many can live in an organism? And then with the we're hunkering down on the gastropods. They're obviously smaller. Like, could they damage the gastropod population and therefore end up damaging themselves in the process, or is it usually pretty cool for the hosts?
Yeah, that's a great question. I don't know if there's probably a limit, right? That means what the limit is probably when the fish dies. But we had some vouchers from Northern Islands from one fish, and the voucher was filled with hundreds of trematodes. Yeah, that was pretty crazy. And I also saw when I was looking under microscopes and stuff, sometimes the sizes of the adult trematodes would be so different. I remember I poured one out into a dish, and it's not in this presentation, but I have a picture of one that's really big and one that's smaller next to it. And so yeah, maybe it is affecting their ability to survive and persist. But I think what makes that complicated is if there's a ton in gastropods or anything. It depends on their host specificity that I mentioned. So if they are able to infect other hosts at that life stage, that will change the outcome of how they're able to get through that.
Cool, thanks.
Any other remaining questions? Okay.
Oh, yeah.
So I had a question about, I don't remember the specific parameter, but it was the one they had to do a lot of estimating for regarding
Dispersal distance.
figuring out dispersal distance.
Yeah.
For that parameter, do you think the fact that the trematodes have multiple potential hosts and you were relying a lot on specifically their fish host would affect your estimation? And if you were to try to get a better sense of that, if you had resources to actually try to figure out full on what that parameter looked like, what would your ideal setup for something like that be?
Yeah. I think, in general, just more attention to parasites in general in research, just because a lot of these assumptions had to be made because we just don't have census data on trematodes, but also, who is infecting who, right? So, when they have a lot of they have, a lot of reef fish that they infect, that could definitely affect their dispersal distance. because I'm only looking, in my data set, I was only looking at Canthurus nigricans. So there may have been other reef fish that we just didn't know had paramphistomes that existed. So it's possible that their, dispersal distance is actually higher, but I would think I would see that reflected in their genetic structure, as long as we have a representative enough sample size. But yeah, I do think having more data, like when people do fish studies, like if they're able to add a component where they get to just identify like parasites that are present, that would be really cool. I think that would help make a lot of this clearer, because those data sets just don't exist.
Thank you.
Yeah.
Okay, so I think that's, I guess I'll come back to the front, sorry.
Talking back, like what time is it?
It's about 4.
Okay.
So I think we can give Randi another round of applause. And then many of you have been to many defenses before, so you kind of know the drill, but for those of you who haven't, Randi is going to go off with her committee to a closed-door session where we're going to ask her lots of really, really hard questions, and she's going to give great answers to them. And then afterwards, we will emerge. She will emerge, hopefully, I'm not concerned at all, triumphantly, a master, and we'll all call her Master Randi. And then we'll have cake and champagne and a little celebration. So it'll be a bit of time, but we'll be in there probably for about 45 minutes, an hour. It depends. We can't tell you exactly. So you just have to wait. I remember when I defended my thesis and my parents disappeared during the closed door session, and then when I came out, people had to call them because they had gone to a bar. And they had to be drawn back for it. But anyway, so we will adjourn to the room. Stay close. There will be champagne, there will be cake, and continue to eat all the snacks that are out here.
Allison, do you want me to bring my computer? Let me take off my mic so you guys can't hear me all mic'd up. No, I don't want anyone to hear that.